Okay, hello to day one, week two, Mr. Olson, BioB lecture. Okay, so today, guys, we're going to be talking about the cell cycle, and then most importantly, we're going to be actually talking about mitosis. Okay, now before we get any farther, let's hit a couple ground things here. These notes that I'm going to go over today are also posted on my website. So if you just want to look at them there, go right ahead. Okay, go right ahead and do it. Now, if you want to have them pulled up or printed off and listen to me, perfect. Okay, that's what I would recommend doing. You're going to learn a lot more that way. This video is probably only be around 9 to 10 minutes. Okay. Now, I am not going to take breaks throughout this video, guys, to stop and have you guys write or anything like that. I'm just going to keep going because it's on YouTube. So guess what? There's a pause button. You can use it. Okay. But that's going to make it just a little bit easier on us. Now today, cell cycle, mitosis. Mitosis, guys, is the first thing in the cell cycle we're going to talk about. What I want to reiterate to you before we go any farther is that mitosis is the replication of cells that are identical to one another. Okay? So think of it this way. When you go wash your hands, which you guys have probably been doing a lot lately, okay, every time you wash your hands, a lot of skin cells fall off. Now, we have to replenish those skin cells somehow. And the way we do that, the way we replenish those skin cells is through a process called mitosis. Now, they are exact copies of each other. And that's important because if you washed your hands, took skin cells off your hands, you wouldn't want hair all of a sudden to grow on top of your palms or fingernails to grow in the middle of your palms. That'd be weird. But instead, it still makes the same exact skin cells that were once there. Okay? Mitosis. Cool? Now, starting off, we're going to talk about the word chromatin. So chromatin is DNA proteins. Realistically, what it is, it's just DNA spaghetti. Oh, wrong way. Oh, let me hide this. It's DNA spaghetti. So you see this image down here? It's just a bunch of mumbled up junk. Okay, this is all DNA. That's great. It is chromatin. That's also great. But what you have to realize here is that this is so messed up and so jungled up, we can't just split this perfectly down the middle. Okay, which is what we're going to get to next. So chromatin. Now, this is also not visible. And it is present during interphase, which we're going to get to interphase in a second. So that word might be a little bit new. Okay, chromatin, though, uncondensed, spaghetti-like DNA. Now, with chromatin, we're going to condense it down. So we're going to take that and we're going to do chromosomes. And those chromosomes are going to be coiled condensed DNA and they're going to be visible during mitosis. So if you think about it this way, if you have a big ball of yarn and you unravel it, the big unraveled ball of yarn is really not that useful anymore if you're trying to go knitting or crocheting. But if it's in a ball and it's organized and neat, now it's a lot easier to use and be able to do what you need to do with it. Okay? So chromosomes are the condensed version. Now chromosomes, guys, are going to be in a shape that looks like this. And if you notice, this shape is a X. Nice. Okay. Now chromosomes are an X, and a half of a chromosome is going to be called a chromatid. And the other half is going to be also called the chromatid. So this chromatid is identical to this chromatid, which is going to be very important in a couple seconds. You're going to figure out why. Okay, identical, identical. They're just connected at something called the centromere, which is just kind of the center point. Okay. So chromatin make chromosomes, which chromosomes split in half and make two chromatids. Okay? Now from there, we need to start at the beginning, the first phase, the first piece, okay? The first piece is going to be called interphase. And inside interphase, there's going to be three other phases. And I know that's a lot, but just how it kind of works out. So the first one is the G1 phase. This is where cells grow, okay? So for us to be able to divide into equal amounts of cells or equal cells, we have to make sure that cell doubles in size. So when we split it in half, it's the same size as the original cell. Okay, so G1 makes it double in size. So when we split it, we have the same size cells originally on both sides. Perfect. The next one is the S phase. This is where DNA replication takes place. So if we need identical cells, we best have the same amount of DNA in that first cell as we do in the second cell. And the only way to do that is to replicate or to double the DNA that's in the first cell split it in half, so then we have the same amount in both cells. And the last one is the G2 phase. Okay, this is where organelles and other molecules are also doubled to make sure that they're split and then put on both sides. So, say that your original cell had one mitochondria, you have to double that, so then your other cell has one mitochondria, and now the other cell only has one mitochondria. Okay. Now, interphase does take roughly 75% of the cell division cycle, okay, which is the majority of the time. Most of your cells, guys, can be either found in interphase or in a resting stage, which realistically is the same exact thing. It just means that your cells are not replicating at that same or at that exact moment, okay? Because if they were always replicating, man, you better be rubbing off your skin all the time. Otherwise, you just have super amounts of skin always building off your body, okay? So, interphase. The cell is ready for division once interphase is over. From there, we start a phase called prophase. Now, prophase is where DNA is going to condense into chromosomes. So we're going to take that chromatin, that unwound yarn or that unwound spaghetti, and we're going to condense it down and we're going to make chromosomes. Now, during the same time, centrioles are going to start moving to the poles. And for you guys to remember, centrioles are the things that help cell division in animal cells. Okay, this is a little bit of review from last unit or last trimester. Okay, and here our image, these are our centrioles. They're spider-looking guys down here. Okay, and now the spindle fibers are going to start forming, which are the legs of the spider. Okay, and then nuclear membranes going to break down. So guys, to get our DNA out of the cell or to split our DNA, we have to make sure we break the safe around it. And breaking the safe just means getting rid of the nucleus. So once the nucleus is gone, then we can actually start splitting things apart. Now, we've went through interphase, we've went through prophase. The next one is going to be called metaphase. Metaphase, guys, is where the spindle fibers attach to the centromeres of the chromosomes. The chromosomes are going to line up along the equator. So you have your centromeres, okay? Take that back. Take that back. You have your centrioles, you have your spindle fibers, and they're attaching to the chromosomes at the centromere, and they're making sure they're all lined up in the middle because they're going to split in half in the next step, okay? But, key trick for you guys. Middle, metaphase. M and M go together. Metaphase, middle, middle, metaphase, metaphase, middle, M and M go together. Cool? Now, from metaphase, we're going to move into anaphase. This is where the chromosomes are going to split and move towards the poles. So our chromosomes that were once here in this last image are now going to split and make chromatids. Okay, chromatid, chromatid. And how this is going to work is that those spindle fibers from before are going to start shortening up. So think of something like fishing. Okay, if you cast out your lure, you have to reel it back in. And for it to reel back in, we have to make that fishing line get shorter and shorter and shorter and shorter until we're all the way back to where you are. Okay, so the shorter the spindle fibers get, the closer those chromatids get back to their original side, the closer we are to getting to two identical cells, or the finish of mitosis. Now, anaphase. Think of the word away. Anaphase, away. Away, anaphase. A and A go together. Okay? Anaphase and away. The next one is telophase. So the sister chromatids are already on each side. Okay, so their chromatids are already split. They're identical to each other. That's why they're called sisters. Okay, so it's like twin sisters. And the new nuclear membranes form. So the nucleus reforms around those sister chromatids. Okay, this way we are starting to actually see that it's going to look like one cell here, another cell here. Eventually, these two are going to split. Okay, now during this phase, guys, the spindle fibers are going to break apart because they're just not needed anymore. And centrals are going to start moving back to their places as well. Okay, and the final step is going to be called cytokinesis. This is where the cytoplasm divides. Cell membranes draw inwards in the animal cell, and the cell plate, or in better terms, the cell wall, forms in a plant cell. Okay, this is just the division of those two cells, so they are now their own individual cell. Okay, one cell, two cell, one cell, two cell. This is the last stage. Okay, now I know that was really fast and there's a lot of it, but here's a little riddle to help you. It's called IP-matic. So we have interphase. We're prepping. We have prophase, we're condensing. We have metaphase, where everything lines up in the middle. We have anaphase, where everything splits apart. We have telophase, and then we have cytokinesis, which is where the cell's going to start pinching in on itself, and we're going to separate into two identical cells. Now, guys, feel free to watch this video again if you want. I'm also going to be posting an Amoeba Sisters video tonight as like your homework. But if you need anything or if you have any questions, please feel free to email me. Okay, we will set up some office hours and we'll start talking about those things, guys, as they are needed. But if you guys need questions answered, please reach out to me. I know this is a little bit confusing, and I know we're only in a 4-day week this week. Okay, so please reach out, please talk to me, all those different things. I'll talk to you guys later. Bye.